For today, what we're going to be talking about is how do we study the prevalence of mutations. So basically, prior to this point, what we've talked about is given raw sequence data, how do I reconstruct a viral genome? And last week, we talked about how can I, in the past 2 weeks. So 2 weeks ago, we talked about how can I align multiple sequences from different viral samples and figure out like where are they homologous, like where do they line up with each other? And then last week we talked about how can I use this to get an evolutionary history. But what if we want to actually know what is the prevalence of specific mutations as they spread across the population? So today is kind of our first step towards how do we actually use all this like genomic stuff that we've done to help with public health, right? Because up to last week, everything has been purely genomics. There's been no public health involved at all. So now we're going to start connecting it to public health. So here's the general outline of today's lecture. I'm going to talk about something called ancestral state reconstruction. I'm going to talk about what's called dating a phylogeny, where basically you're converting it to unit of time. And then I'm going to talk about assigning genomes to lineages. And I'm going to end with how do we actually study the prevalence of mutations. And that's really the core public health part. So the first three are, again, still more kind of bioinformatics questions. But then that last part is really the public health connection to all this. So first, what is ancestral state reconstruction? And to recap, remember that we can think of the evolutionary history of some ancestral sequence down an evolutionary tree. So basically along these branches, maybe along this branch here, this G turned into a C, and then kind of as we go down, we have mutations that accrue. And then in the present day, the only sequences that we can observe are the sequences in present day, which represent the leaves. So the tree and all of the evolutionary history is completely hidden from us. We can't see it. And last week, we talked about what's called phylogenetic inference, which is a way of given just the modern day sequences, assuming we have a multiple sequence alignment, which we've already talked about how to construct. But yeah, given just the modern day sequences, we can estimate this evolutionary tree, right? That was last week's topic. But the question is, how do I actually reconstruct this mutation history, right? All I got from last week is the tree shape, not the history of the mutations. So how can I use that information to actually reconstruct this mutation history over time? And that's kind of the goal of today, or this first part of ancestral state reconstruction. So what is maximum likelihood ancestral state reconstruction? So first, it turns out that we can use an algorithm that's very much like Felsenstein's tree pruning algorithm to reconstruct those prehistoric mutations that happened back in time. So remember Felsenstein's algorithm, I didn't go into the details, but I just mentioned that we can use Felsenstein's tree pruning algorithm given a tree and sequences and a model of evolution to compute a likelihood score, right? That's what we used last week to come up with our maximum likelihood tree inference algorithm, where given a tree, sequences, and parameters, I can compute a likelihood score using Felsenstein's algorithm, and then we just kept changing what tree we had and keep computing the score using Felsenstein algorithm until we got a maximum likelihood, one that maximized that score. So we can use an algorithm very similar to it to reconstruct those ancestral states, those previous in history mutations. So basically what we would do is essentially just a post-order traversal of the tree. So if you haven't taken computer science classes, that's totally fine. Basically, a post-order traversal is just a traversal over the nodes of a tree, where basically we're guaranteed that we're going to look at any given node after we've already looked at its children. So basically, we're going to process the leaves first, then we're going to process the parents of the leaves, then the parents of those nodes, then the parents of those nodes, all the way up until the root would be the last node that we visit. So basically, we're guaranteed any given node that we're looking at, we've already looked at its children. So how do we do this, this ancestral state reconstruction? How do I reconstruct the sequence at every given node in my tree? If my current node, the current node that I'm looking at in this post-order traversal, if it's a leaf, I already know its sequence. sequences, right? By definition, the sequences in my multiple sequence alignment are the sequences of the leaves. So for the leaves, I already know its sequence. I just pull it from the multiple sequence alignment. Otherwise, so for any internal node, any node that's not a leaf, for each position of the sequence, I try to find the most likely symbol based on the symbols of the children. So for example, if all of my children have an A in that position, then I'm going to guess that the most likely symbol for me is also an A. Versus if I have like, maybe a lot of my descendants have an A, but a couple of them have a C, maybe I would think that the most likely one is still an A, but I'm less sure now. So I can basically compute a likelihood score for each position of my sequence based on the likelihoods of each of the letters of my children's sequences. And then the result is I can basically label every single node in my tree with my best guess of what its sequence is, right? So for my leaves, I know exactly what it is. Every other node, based on my descendants, what is my best guess at what the sequence is? And the specifics of the algorithm are a little bit out of scope for this class, but if you're interested, feel free to reach out to me and I can send you a link. But it's a fairly simple algorithm, if that kind of makes sense. Any questions about this concept? Any concerns? No? Okay. So I can basically feed that algorithm my tree and just give it my leaves. And each internal node, I'm going to predict what is the most likely sequence based on my descendants. And then at this point, I've reconstructed the full evolutionary history of the virus, including where exactly all the mutations occur. Okay, any questions about that? No questions? Okay, so the next section is called dating a phylogeny. And we don't mean dating in the sense of like social dating, but we talk about dating in terms of determining the date of something. So converting it to time. So when I inferred a phylogeny using the algorithms I talked about last week, the branch lengths are a unit of number of mutations, right? The models that we come up with, they're basically just determining how many mutations exist between this node and this node, but we don't know how much time it was. We might know it's like 5 mutations or something like that, but how much time equals 5 mutations, right? So we know how many mutations occurred from parent to child. However, for public health, it would be a lot more helpful if the branch lengths were unit of time. So maybe rather than 5 mutations from parent to child, maybe 2 days or whatever unit of time would describe the relationship between parent and child. Because then what I could do is take the entire evolutionary tree and kind of track down how it spread over time, maybe overlaying other information. And it would tell me how exactly this virus spread across the world over time. So what I want to know is how much time has passed from parent to child. So remember, I can infer the mutation phylogeny, like the phylogeny where the branch lengths are unit of mutations, just using the sequence. That was the subject of last week's talk. But if I also knew time information, so for example, let's say that I have a bunch of sequences that I collect from a bunch of people across the world. I have a timestamp on each of those sequences. I know exactly what day that person came into the clinic. So I know exactly what day I collected the viral sample from, like maybe a nasal swab or however I got it. So the goal is, given that I know the timestamps of the leaves of my tree, because I collected those, I know exactly when I collected that sample, I can rescale the branch lengths of my tree to kind of maximize the matching of the branch lengths with respect to those time scales. Does that kind of make sense? So I have hard timestamps on each of the leaves, and I'm trying to rescale all the branch lengths such that those timestamps fit as perfectly as possible. Okay, so what I'm essentially doing is estimating a mutation rate for each branch. I'm basically estimating the number of mutations per time along this branch is some number. So I know the number of mutations along this branch. Now let me rescale it by the mutations per time to now rescale it to be in unit of time. Okay, so this algorithm, like the way to do this type of rescaling, the way to date a phylogeny, it's not very simple. There's a bunch of complicated techniques that try different approaches to do it. But fortunately for us, we don't have to worry about that. We can just take our pick from existing tools and just use them kind of as is. And I won't go into the details of how each of these tools work because they each use kind of different, pretty non-trivial, like sophisticated stuff to do it. But I have links to them as I display them, so you can always look into them if you're curious. So one of the more popular tools that people use is this tool called tree time. And you can find the source code on GitHub. It's written, I believe, in Python, if I recall correctly. So this is kind of the most popular tool right now. And a bonus is that it actually does both. It can do scaling your tree to be dated, and it can do ancestral state reconstruction. So it's kind of a nice benefit that it can knock out both of those at once. Another popular tool is called LSD2. So LSD stands for least square dating. It's not what you might think LSD stands for. But it uses an approach called least squares method to determine those branch length mutation rates. And LSD2, you can find it on GitHub as well. I don't remember what language this one's written. I think it was C++, but I'm not 100% sure. And then a less popular tool, but that's also of interest. So this is a tool that came out from one of my colleagues pretty recently. It's called LogDate, that it also uses a completely separate approach for doing this type of dating. And you can find that on GitHub. I think that one's written in Python as well. So yeah, so I won't go into the nitty-gritty details of how each of these tools works because each of them is making different assumptions and using those different assumptions to do a different computational approach. But if you're curious, feel free to reach out and like also feel free to look at their code bases and it might make more sense. Okay, any questions? Okay, so up to this point, what have we gotten to do? So last week, we figured out how to get the tree. After this first bullet point, using the tools that we can describe here, I can reconstruct the mutations of those past ancestors that I no longer have access to. So I can actually reconstruct the full mutation history. And then as of the second bullet, I can also scale the branch length to no longer be in terms of number of mutations, but actually be in unit of time. So now I'm getting some pretty helpful stuff that maybe is going to be helpful down the line for public health inference. But the last thing that I want to talk about before we go into the public health aspect of it is how do we assign genomes to lineages? So a lineage, just to kind of clarify the terminology, a lineage in terms of evolutionary biology, it's just a subtree of the overall phylogeny, right? So I think with the COVID-19 pandemic, there's all these lineages coming up like B117 and whatever. A lineage, just generally speaking, is one subtree of your overall phylogeny. That's all it is. So biologically, it's basically given some subtree, a lineage is all viral sequences that inherited all the mutations up to that point, right? Because some node has all these mutations up to that point. The lineage is everything below it that therefore inherited all those ancestral mutations. A strain, it's similar to a lineage, but it's specifically a significant deviation from the ancestral virus. So it's differentiated so far enough that it's no longer considered a lineage of the same virus. And you'll notice I'm being very hand-wavy with how I'm describing it. And it's because, so my background is actually not in public health. I'm a computer scientist. But I tried looking up what exactly is the definition of significant deviation. Like when exactly do we start calling something that used to be a lineage a strain? And I couldn't find a firm or specific definition for what was considered significant. So the best that I could find is that it seems to generally just have some implications about significant functional or phenotypic differences. So this term phenotypic, if you haven't taken a bio class, you might not be familiar with it. Phenotype is just the physical or like the like the physical structure, like the physical visibility of your genes. So genotype is your actual mutations, your genomic sequence, and phenotype is how does that manifest in some physical or functional difference. So yeah, so even this is kind of hand wavy, and I wish I could have come up with a more firm definition. But just basically, somehow it means it's a lineage that has kind of evolved so far away from the ancestral virus that now it has some significant functional differences. Maybe vaccines don't work the same because one of its proteins has mutated significantly, or maybe it's a lot more transmissible, or something like that. So for SARS-CoV-2, people use a tool called pangolin to assign genomes to lineages. So basically, we run through that workflow to go from raw data to a genome sequence, and then people feed that genome sequence through this tool called pangolin to assign it to a SARS-CoV-2 lineage. And pangolin, you can access the source code on GitHub. I think it's written in Python as well. Yeah, so this is kind of the current state of the art of how people do this assignment. Any questions about this slide? Any questions about the terminology? Any confusions, concerns? Kind of makes sense. Okay. Okay, if you have any questions, feel free to type them in the chat. I'll keep going, but like, please feel free to interrupt. Okay, so that's kind of the bioinformatics background of today's topic. So we figured out how do we re-estimate those like ancestral mutations that occurred. We figured out how do we actually scale an evolutionary tree to be a unit of time. And we figured out how do we assign a given genome to a lineage. And the way that pangolin works, I don't want to go into the details because that's also kind of a little bit too technical for this talk. But if you're curious, feel free to check out the source code and feel free to hit me up and I'm happy to answer questions about it. But okay, so now we're assuming that we've assigned every single one of our genome sequences to a lineage. We assume that we have the full ancestral like mutation history and all that good stuff. How do we actually use all of this data that we've generated to study the prevalence of mutation? And how can we come up with public health actionable information? So I kind of just want to give an overview of some of the popular tools that people use. And if you want, after I showcase them, maybe I'll do a little bit of a demo. So NextStrain is the most popular tool right now for tracking viral mutations. So basically what NextStrain allows you to do is it lets you visualize the phylogeny plus the mutations plus demographic data about the samples, which is super powerful because once you put all of these things together, you can start really deconstructing the history of the epidemic. So here's the link to NextStrain, and specifically this is the NextStrain build for SARS-CoV-2. Originally, NextStrain was created, I think the original term was next flu, and it was meant specifically for influenza. It was to try to like predict the seasonal influenza spread. But then they repurposed it during the COVID-19 pandemic. Well, I guess a little bit before COVID-19, they repurposed it from next flu to next strain to just be generally applicable to viruses. And it became especially important during COVID-19. So just real quick, this is kind of a screenshot of how it looks like. But since we have plenty of time, I'm just going to maybe like pull it up and we can play around with it. So if you go to that link, it'll take you here. This is the NextStrain website. You can also just Google NextStrain and it'll show up. I'm going to click on the latest SARS-CoV-2 analysis. And it takes a little bit of time to load. And we get this pretty looking tree. But what does this actually mean? So this is an evolutionary tree where this is back in time, this is forward in time, and this is actually a dated phylogeny. So you'll notice that the branch lengths have been scaled such that the time axis, like the distance from the root to any given node is time, right? And as a result, because I know the date at which each sample was collected, I can basically scale the tree and align it to have dates associated with each of the internal nodes. So let me just highlight one of these leaves real quick. So this one, for example, this is a sample that was collected in Ecuador, and we have information of some of the interesting mutations that it has. And we know, so the divergence, I think, is the number of mutations from the root, if I'm not mistaken. Let me double check. Yeah, seems to be. We know the collection date, and then the clade, that's the lineage. So here it says 19B. That doesn't really mean anything to you and me, but for virologists, that tells you like what viral lineage does this belong to, and it has information about the author. I can click it to have a little bit more information. So here is a bunch of metadata from this sample. And I can click out of it to make it go away, I hope. Yep. Whoops. So what's cool about this is you'll notice just by doing this, it tells us a lot of information about how this virus spread across this population. So this one here, I'm currently coloring it by clade. So it tells me that this like purple clade is closer to the ancestor. And then what branched off of it was this like dark blue clade. And then and by clade, clades are just lineages. And what branched off of that is maybe this like light blue lineage. And then off of that, this green lineage branched off. And like it kind of, you get this clean history of how the lineage is passed over time. And all they're doing is just taking the evolutionary tree, and they know the lineage assignments for the leaves because they can run it through Pangolin or some similar tool. And then all they're doing is just painting each of the internal nodes as some proportion of how many of its descendants also had that label. So the label happens to be the lineage, but I could pick any label. And you'll see here it has this color by. I can pick anything I want. So let me pick country. And in this, it's not as obvious to see what we're looking at, but basically here we don't really have any confidence in terms of like what country it came from back there, because they've like subsampled this quite a bit. But like we'll see, we have this big cluster of red things, which red, let me move my zoom stuff. Red seems to be USA. So we have like this cluster of sequences from USA that seems to have branched off from something over here that, you know, it looks like we got like Paraguay and I don't know. We basically get some sense of like how it's spreading across different countries. And I can switch it to region. What are these regions now? Region North America versus Asia. So this is a little bit easier to interpret. Where here we have like the ancestral ones are from Asia region that then kind of branches out to North America over here. But then later it separately branches out to Africa, and also it separately branches out to Europe. And we see here that the bulk of our North American sequences actually branched off from these European branches. So this is super helpful because it tells us, you know, kind of at the beginning of the pandemic, there was all this question of, you know, should we restrict travel? And if so, what country should we restrict travel from? And you'll notice that the government actually restricted travel from Asian countries, but the USA dominant viral, like if you actually looked at the evolutionary history of the viruses, it seems like the branching of the viruses that we were getting in the USA actually came from Europe. So what's funny is if they had done this type of simple, just eyeballing analysis, they could have come up with much better informed policies. But just, you know, generally speaking, it's clear that you can come up with these types of actionable pieces of information from this. Yeah, so they have quite a few other features. You can like change the branch lengths to be in unit of mutations rather than unit of time. So here now divergence is what they call mutations. So the ancestor has 0 mutations from itself, but then this is like 5 mutations away from the ancestor, 10 mutations away from the ancestor, so on. Pretty cool stuff. Cool. And then scrolling down, we actually have a lot more information. They show us like the prevalence of it across the world. they show us the genome of the virus. So this is like the different genes of the virus. We have ORF1A, ORF1B, the spike protein, kind of the other parts of the genome. And here we can see what is the diversity, what are the kind of like numbers of different mutations. It's shown in entropy. If you haven't taken information theory, you might not be familiar with entropy, but this is basically like the disorder, the less uniformity. So the more deviations there are for every given position. So we'll notice like this position, we have a bunch of different nucleotides, or I guess this is amino acids. And like here, we also have a bunch of different letters at that position. But then other positions are a bit more conserved. Like they have lower entropy, lower disorder, so they have a lot more conservation. Like all the sequences are pretty much identical here. So you can kind of look at this type of information. So like this is helpful for if you're trying to come up with a vaccine, for example. Because if you're coming up with a vaccine, you want to pick a part of the genome that basically doesn't mutate too frequently. So if I'm designing a vaccine to target the spike protein, just eyeballing based on this, maybe I want to pick a region of the spike protein in this window to design my vaccine against. Because this window doesn't seem to mutate very much compared to like these other windows maybe mutate much more. And if there's more variability in this window, that implies that my vaccine might not end up being as effective across people from different populations or over time. So, you know, a bunch of interesting stuff that we can come up with just by looking at this plot. And then here we can see the frequencies of, let's see, is this frequency of each region? Yeah, but I can change this to like Clayton maybe. So here we can see the prevalence of each of the lineages of COVID-19 over time. So it seems like at the beginning, this 19A lineage was the dominant one. And then over time, 19B kind of also died out pretty quickly. 20A picked up, 20B picked up. But then over time, those ones seem to like die out. And then this one is kind of the dominant lineage right now. So we can see how are the different lineages spreading in prevalence over time. And then here they just have some like information, blah, blah, blah. So yeah, it's a super cool tool. Definitely like take some time to play. Oh, I forgot about this feature. So you can actually click play and it like moves forward in time with respect to the evolutionary history and also simultaneously with respect to the map. So like the map is actually showing how the different lineages or whatever demographic information you pick to color by, how are they changing in terms of frequency in different countries over time. So like here we see the green one is the most prevalent in the US, but let's see if that changes over time. So, oh, the green one is starting to decrease in the US. Oh, it's going back up. But now it's starting to decrease, going back up. Those things like, oh, now this yellow one's starting to pop up. So yeah, stuff like that, it's just like super wild what you can do. And yeah, the hope is you can motivate public health intervention using this type of real-time information. Any questions about this one? Any questions about NextStrain? Like definitely feel free to just like play around with it. This is all public. It's all, you know, you can access it from your web browser. Oh, there we go. The yellow one's like taking over in the US. Yeah, just it's really cool visualization stuff. Okay, so I'm gonna jump back to the slides real quick. Make that full screen again. So yeah, so the link to NextStrain is in my slides, but if you just Google NextStrain, you'll probably be able to find it. Whoops, that's not where I want to go. There you go. Okay, so that's kind of what it looks like. So another tool, so NextStrain is kind of the most popular one that people use, but there's another helpful tool called Covariance that I think is built largely by the same team. But basically, it specifically is meant to help you track the COVID-19 variants and mutations over time. So for this one, just for the sake of time, so I'll show you a brief screenshot. This is kind of how it looks, but we have plenty of time, so I'm just gonna open it up and play around with it. So Covariance, you can if I want to look at it per variant, I can click here. I don't like the terminology of variant that's going on in, and I guess so the terminology of a variant in the context of viruses is a collection of mutations, and that's slightly different from lineage in that with viruses, because the genomes are so short, you can have multiple separate lineages give rise to the exact same collection of mutations completely independently. So this is a concept known as convergent evolution, kind of like bats versus like bugs, like flying bugs. Both of those things have wings, but those are not homologous wings. They both evolved wings independently of one another. So two completely different lineages had convergent evolution. Their evolutionary kind of like the fitness that drove their evolution independently converged upon, oh, I should have wings. So similarly, like lineages, if there's specific mutations that are beneficial for the fitness of the virus, maybe it helps it invade more easily or something like that. Different lineages of the virus could independently evolve those mutations, right? Evolution is just random mutations happening, and different lineages could independently end up with those same mutations. So a variant in the context of virus is like it's not really a lineage because they could have given rise independently, but it's defined as like a set of genomes that have the exact same specific special mutations, if that kind of makes sense. So mutation is like an individual letter that changed, and a variant in the context of a virus is a collection of, like, it's kind of like a cluster of the genomes that have the same exact interesting mutations, whether they evolve them together or if they evolve them independently, it doesn't matter. And that's not what variant means in other genetics contexts. So that's kind of why it bothers me. But anyway, but yeah, so here we can like play around with this type of stuff too. So horizontal axis here we have time, and then each of these curves is one country, and we can see like how was each lineage. So this is the lineage 20E. How prevalent was it in each of these countries over time? Right, so we can play around with like different lineages broken down by country. Or what if I want to do the other way around? What if I want each graph to be a country and I want the curves to be different lineages? So maybe I'm interested in the US, like what lineages were prevalent in the US over time. So it seems that like by this point, the like dark purple one was the most prevalent. Oh, actually gray. Gray is others. I don't know what that means. I guess just arbitrary. I don't know. But yeah, so this like dark purple 20C was the most prevalent single lineage. But then over time, this like dark red one, this maroon one, 20I or whatever it is, overtook. So I don't know. It's just kind of like cool visualizations that you can see over time. Nifty stuff. Okay, so that is covariance. So similar to NextStrain, but it's specifically focused on those variants and how the variants are spreading over time. Whereas NextStrain is more focused on the evolutionary history overall and like the mutations as they occur over time. And then the last one that I want to highlight is a tool called Outbreak.info. So Outbreak.info, it's a tool developed, I think, primarily by the Anderson lab. So Christian Anderson is a professor, or he's a researcher in Salk Institute. Is he at Salk or Scripps? I'll have to double check. But basically, Outbreak.info, it tracks the cases, deaths, and lineages in specific populations for COVID-19. So let me actually double check. Yeah. Salk or Scripps? Anderson lab, Scripps. Whoops. I should know this. Yeah, so it's created by the Sioux lab, the Wu lab, and the Anderson lab at Scripps Research. And like, let's say I want to look at San Diego. So this is cases and deaths. So I go to San Diego, and here I can see the number of daily new cases in San Diego over time. So every day, how many new cases there were, which this is looking nice because it seems like we're getting to the stable point, which is good. If I wanted to look at like daily new deaths, I could do that as well. And we see a similar trend, and deaths generally lag behind the cases plot, but also it's going quite down, which is nice. You know, daily number of, oh, we don't have test information. Interesting. I don't know, cumulative deaths. I don't know, just useful stuff to play around. You can normalize by the population. Is this over? Oh, this is 100. Yeah. Interesting. So yeah, that you can play around with the cases and death information. If I want to look at the variance, I can track the variance in San Diego over time. So here is that similar plot that we saw on covariance, but now we have like fine-grained, like county level, right? This is all San Diego. I don't know if it's San Diego City or San Diego County, but either way, we have like city or county level rather than covariance was only country level. And yeah, we can play around with, you know, like how prevalent was each of the different lineages over time. And then we can break it down by specific mutations. So these are like different mutations, like actual mutations of the genome sequence that might be clinically relevant. And we like highlight them of like variant or mutation of concern versus variant or mutation of interest. I don't know how they define concern versus interest, but you could probably find that in their like about page. I could probably click on the mutation of concern. Does it describe why this is concerning? Huh. Oh, okay. Well, I guess they have a bunch of, so you can just click on it and they have a bunch of papers that describe it. So if we wanted to learn like, why is this mutation concerning, we could click on a bunch of these links just to get some better info about that. And then let me go back to the home page. And then the last part is research library. So like, what am I looking for? Maybe I'll search for that. Do I find anything? Let's see. I thought I would find my paper. Oh, 10,000 results. Dang it. I'm gonna look up. I'm gonna cheat. Wait, what? Oh, there we go. So this is mine, this is mine, this is mine, this is not mine. Where's my other papers? Whatever. But yeah, you can like look up research. And then if you're interested in programming and you want to build your own tool that kind of utilizes these data, they have an API. So for people that aren't familiar with computer science, an API is just basically a generic usage description for how do you query some data set or some database of some sort. So they have descriptions of, like for the COVID-19, yeah, so you can kind of like look through this and figure out how do I utilize their API within my own tools that I want to write. How do I find usage details? See document structure? Interesting. So I don't really know. I guess maybe there's documentation somewhere. But yeah, so you can figure out how to query their database using your own tool if you want to like wrap it into some other tool that you have. Oh, okay, they do have documentation. Nice. So I can click here. Wait. You may refer to the mychem.info API documentation. Okay, nice. So yeah, if you want, you can always look at this to see like, how do we query this database? And I guess this one is identical to it. Okay, so let me go back here. So these are kind of the main tools that people are using right now. I just wanted to give a brief overview of what they are. Um, but yeah, if you're interested, feel free to play around with them, all that good stuff. I think that's all I wanted to present for today. But one kind of lasting message that I want to leave on is, you know, the past weeks 2 through 6, right? Because I think this is week 7. Yeah, so weeks 2 through 6 was all focused on like more algorithmic and more like computational problem solving. But I want to emphasize, so I myself, I'm a computer science person, and like the stuff that I work on is primarily in the computer science space, like developing algorithms, developing tools. But what's really important is figuring out how to make your tools easily accessible to the non-computational, like public health official or like epidemiologist, or importantly to the general public as well, right? Like you don't want to have someone needing a PhD to be able to understand what it is that your tool is showing. So having some like intuitive visualization tool that anyone in the world can just click on and play around with, and the visualization is intuitive such that without any significant prior background, someone can just kind of figure out what it means. It's a really important problem. Um, so in the computer science world, this is largely in the space of what's called human computer interaction, or HCI. Like, how do we design interfaces that are very intuitive for a user to play around with? But just kind of like one overarching um, like message I would have is, if you ever choose to go into developing these types of tools, or like developing any types of bioinformatics tools for public health, make sure to also think about how is it going to be useful in the hands of someone that doesn't have super technical expertise.